for R&D activities, then capacity building, as well as human resource development in frontier areas of biotechnology and allied sciences for any region. And for human resource development, we used to conduct 7 days training program, 3 training per year for the faculties and the PG and postgraduate students. And for we have training that is known as outreach training program. It is conducted at the field level. So that is one this program at field level. Lecture series. Multiple lectures are planned, and eminent scientists of different background within and outside the country, they are invited for offline as well as offline lectures. Lecture of this series is going to be delivered by Dr. Kumar Chaudhary, Assistant Professor, Guru Veterinary and Animal Sciences, University, Ludhiana, on the topic stem cells and our speaker. So I'd like to request our Director of Research to say a few words.
Thank you, Dr. Deka. Deka, the professor, Associate Professor in charge head Department of Animal Biotechnology and the project coordinator of the DBT funded state level biotech hub. In the department and also in the university, those who are participating in this particular platform for today's online lecture, the project staff of various projects being implemented under the Department of Animal Biotechnology, the respective participants joining this platform today, and more particularly, the honorable speaker who have been invited today to deliver an important lecture on a very, very important topic, Dr. Ratan Kumar Chaudhary from Gadwasu, Ludhiana. So this is really a privilege for me to be a part of this program today, to listen to the lecture to be delivered by Dr. Chaudhary on a very important topic, not only in terms of veterinary research and therapeutics, but also it is a general topic of interest because stem cells are the cells for the future to develop regenerative medicine as well as the therapeutic for use both in human as well as veterinary medicine. So this particular lecture series online have been designed under this phase 2 of the DBT funded Advanced Level Biotech Hub program. And we are fortunate that since beginning, this department has been hosting the Advanced Level State Biotech Hub for the state of Assam. In reality, the entire proposal for Biotech Hub establishment in the Northeast under the Department of Biotechnology, Government of India was initiated and conceptualized in this particular department. And as per the proposal submitted to Government of India, it was materialized in the entire Northeast, and 126 biotech hubs were established in 8 different states of the Northeast. Among these, 66 biotech hubs were established only in the state of Assam. And this institute, the Advanced Level State Biotech Hub hosted by this department was the nodal center for activities under the Biotech Hub funding of DBT for the entire state of Assam. So as it has been rightly pointed out by the project coordinator, Dr. Deka, that this is mandated with the capacity building, the training, as well as research in emerging areas of biotechnology. And as this institute is specialized for research in advanced areas in livestock and poultry, that is the veterinary and animal husbandry research. So the research project that has been undertaken under this State Biotech Hub also is associated with the research in the front of application and development of biotechnological tools like diagnostics and vaccines against some important prevalent viral diseases of swine. And as a part of the entire program of capacity faculty development, this online lecture program has been initiated, and we are happy today that one eminent speaker, Dr. Ratan Kumar Chaudhary, who is an assistant professor in Department of College of Animal Biotechnology in Guru Angad Dev University of Veterinary and Animal Sciences, Ludhiana, have kindly consented to be the speaker. So as we all are aware, the importance of stem cells and their uses for regenerative medicine as well as for stem cell therapy in both in human and animal medicine. This is a very, very significant topic, and we are hopeful that through these deliberations, the participants and students across different institutes who have joined today in this particular online program will be benefited. So we are eagerly waiting to listen to Dr. Chaudhary on this important topic. So I'll not lengthen my speech, but I'd like to express thankfulness to the funding authority, the Department of Biotechnology, for generously funding this institute under various programs, particularly under this Biotech Hub initiative, constantly for the last more than 14 years, since 2010. So I think this has benefited a lot, not only to this institute, but also many other institutes of the state, as well as the entire region of Northeast India. So with this, I welcome Dr. Chaudhary for his deliberations. But before that, I think Dr. Deka will introduce him to the audience. Thank you.
Thank you very much, sir. Before introducing Dr. R.K. Chaudhary, I'd like to thank our Director of Research because he was our head of the department in the animal biotechnology, and due to his pioneering effort only, this ALSBT Hub project, second phase of this project has been operating. Really very much thankful to him. And in the last December, he joined as Director of Research. Since then, I've taken over as a Dr. Ratan Kumar Chaudhary, I am delighted to introduce him because we were colleagues when I was working in Dr. Chaudhary completed his BSc from Tamil Chennai, and then he first program in the animal genetics and breeding from IVRA, Verily. So he completed his PhD in animal science from the University of Maryland, USA. Then he worked there for 6 years under the USDA facility. And he also pursued 2 postdoctoral research. He got his PDF postdoctoral fellowship from University of Kentucky and University of Vermont. And he has been awarded the Best Teachers and Best Researcher in 2023 from the College of Animal Biotechnology, Ludhiana. And his first area of work is on the regenerative medicine, memory stem cell, and veterinary diagnostics. So he has authored, he has edited 3 books which have been published internationally. And one of the example of this book is Stem Cells in Veterinary Science, published by Springer. And he also developed 2 mobile apps, including Stem Cells, Basic Understanding and Clinical Applications, available on the Google App Store. And he has published very quality articles, more than 60, in the reputed national and international journals. And he has written 10 book chapters and more than 60 presentations. And he also could establish an international collaboration in stem cell research and applications in the country. And in Google Scholar, so he has more than 600 citations with H index of 15. So in my knowledge, he is really a very sincere and very hardworking guy, a very kind-hearted person. So really, I'm happy to see you here again. So Dr. Chaudhary, it's over to you now. Let's start.
Thank you, sir. Thank you to the Director of Research of Assam Agriculture University, and also thank you to my senior, Dr. Deepak Dekar, who has invited me and provided me an opportunity to talk about my research and also to introduce you um, about the stem cells, how this stem cells, which is a unique type of cells present in the animal's body or you know within us also, and how this can be utilized to enhance the lactation in bovine and also to treat mastitis. Okay. So let me share my presentation. So what kind of audience do I have here, sir? All are student, or it's a mix of student and faculty, and
Yes.
also the veterinarians, field veterinarians?
Yes, faculty, students, and students are, some of them are from general background, and majority of them are from veterinary.
Okay.
Some of the faculties they are also joined.
Okay.
Other department, yeah.
Are you able to see my full screen? No.
Not yet visible.
I don't know. Here it's happening, but it's not happening there. So let me do this way. Ah,
Try again.
You can stop presenting and try again once again.
yeah. Okay. Yes. Now it's okay, right?
Again, the same thing, say black screen is appearing. So are you selecting the entire screen?
Yeah. This time I am taking the entire screen. Last time I selected a window.
Even then it is appearing as a black screen only. Nothing is visible.
Is the same? No change?
No change, no change.
So did you open two connections here? No.
In the same room, have you opened two different systems?
Yeah, I have a dual screen. So let me disconnect to one.
You should close one, yeah.
Yeah. Okay.
That will be issue. Again, you should stop sharing and again, you should try again.
Okay.
It is in the process. Let us see.
How about now, sir?
Oh, nothing has come.
Sir, but it is visible to us, sir.
Yeah.
Okay, you can continue.
Okay. So the topic is stem cells and enhancing lactation persistency and combating mastitis in bovine. As you all know, I mean, Dr. Dekar and DR sir also introduced me nicely. I'm in charge of Animal Stem Cell Lab at the College of Animal Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University. I have been working here since the last 11 years, and this is who we are. So my lab is working on mainly on stem cells, and apart from that, we are also working on regenerative medicine in dogs. Currently, my lab has two projects, and that's we are working on stromal vascular fractions that is derived from the adipose tissues of dog, and trying to see therapeutic effects on the surgical wounds in canines. We are also working on detection of early pregnancy in buffaloes as early as 20 days. And we have developed this technology to detect the early pregnant buffaloes. And we are also working on finding an alternative treatment of bovine mastitis using stem cells and its derivatives. And to some extent, when Dr. Malik was working here as a dean, we also, our lab is engaged in veterinary diagnostics, working on some of the viral diseases and develop some diagnostic kits. So, but basically this lab, animal stem cells lab, is focused and highly engaged in finding or working on stem cells of veterinary importance. Okay, as Dr. Deka said, we have developed a mobile app. So those who wants to learn more about stem cells, I would encourage scan this code and download the app to know more about stem cells, its applications in veterinary medicine. To some extent, you also find its applications in human medicine, because this app has been developed in collaboration with the USA collaborator, Asymmetric LLC Boston. Okay, so before we go into the detail and understand what is mammary stem cells and how this could be utilized as a therapy, let's start with what is stem cells. Okay, so stem cells are any cells that are blank in nature, not differentiated, not having any specific functional properties. They are termed as stem cells. Okay, and these are the properties able to self-renew itself, able to give rise to many different cell types. That's called multipotency. Plasticity, it can change into different types of cells. Say, fat cells can become memory cells can become neural cells, and so on. It has an intact genome integrity, means they doesn't allow any mutations to happen in their genome. Transplantability means if you inject those cells into the mice, or it will able to produce an organ-like structure. Say if you take the liver stem cells, it will produce the mass of a liver inside the kidney capsule. If you take the any other cell type, that will make those kind of tissues within the animal. So that means it's testing the self-renewal ability and differentiation ability of those cells. Okay, it's a niche dependent. Niche dependence means if you take those stem cells out of their environment or microenvironment, they tends to change its behavior. They may tend to differentiate. Okay, so niche is very important. So these are the 5 or 6 important criteria that fits into the stem cells. I'm giving one example of stem cells, though there are many different types of stem cells. If you start from here, then from the sperm and oocyte forms the morula and blastula, and then from a morula you can give the naive kind of embryonic stem cells, and then you have a blastula that from the inner cell mass you can have the embryonic stem cells, and you can go on. So, and these stem cells can be of different types in terms of potency. Okay, so highest potency goes to the totipotent, and then goes to pluripotent, multipotent, oligopotent, unipotent. Okay, and also we have a cancer stem cell, and then finally you have a induced pluripotent stem cells that you all know. So out of these different cell types, there is also called adult stem cell, or more specifically, it is a tissue-specific stem cells. So each organ or tissue has a stem cell population. They are called the tissue-specific adult stem cells. or tissue-specific stem cells. So one example which is vastly studied tissue-specific stem cell is the hematopoietic stem cells. So hematopoietic stem cells, you have a common myeloid progenitors that give rise to RBCs and then give rise to platelets and also give rise to different cell types like neutrophil, basophil, eosinophil, and monocytes, okay? And then so on, plasma cells, memory cells. So you can see that one hematopoietic stem cells from here can give rise to different cell types within the tissue. So therefore the blood is the tissue. Okay, so if you talk about other stem cells like, muscle, so you have muscle stem cells, cardiac stem cells, neuronal stem cells, intestinal, liver, memory. So all the different tissue types, they have a specific stem cells, and they together called tissue-specific stem cells, okay? Okay, so today we will talk about only one type of adult stem cells, and that is the mammary stem cells. So remember that understanding different cell types is very, very difficult, because this mammary stem cells scientists are studying since the last 60 years. Still we are clueless. We can say it's a more puzzle than the explanation. Okay, so the mammary stem cells in the mammary glands are responsible for growth of mammary gland, tissue homeostasis, tissue turnover, especially during the lactation. It replaces and regenerates the mammary epithelial cells, and this is the cells which secrete milk. So in other way, more number of epithelial cells is proportional to more amount of milk. So the cow having a big udder and a huge number of epithelial cells will give rise to more milk in compared to the cow having less udder and less number of epithelial cells. So the number of epithelial cells is directly proportional to the amount of milk, which is shown here. And also the secretory activity per mammary epithelial cells. So there may be mammary epithelial cells, but if those cells are not functionally differentiated, or they are not secretory in nature because of some hormone imbalance or maybe because of nutritional imbalance, then those epithelial cells will not secrete milk, because first is the maintenance of the tissue or the organ, followed by the lactation. And recently, these stem cells being used for regenerative medicine, especially mastitis treatment. Okay, so you all know this is the anatomy of cow mammary gland. So it has a left rear and right rear, and in the front you will have a left front and right front. So each cow composed of 4 quarters or 4 mammary glands. And so in US, the killing of cow is allowed. So all the work which they perform on cow, they can slaughter, take the whole udder out, take the tissues out of it, and measure different parameters which is relevant to the product. Unfortunately, it is not in our case in this country. So this is this structure which is called mammary parenchyma, and this mammary parenchyma is composed of again different cell types. Okay, lymphocytes are there, neutrophils are there, and capillary fibroblast cells, mammary epithelial cells, again that's is surrounded by myoepithelial cells, and then you have a basement membrane, and then collagen fibers. Okay, so what I'm showing here is the mammary epithelial cell is one of the cell present in the mammary gland, So there are 9 different types of cells. So some of the many experiments have been done using, taking the tissue of the cow or say goat, and considering that this is a transcriptome of whole memory epithelial cells may not be the right answer, because this is, so what the transcript you get from those tissue is a transcript of all those 9 different cell types which is present in the tissue. So that's not a true representative of memory epithelial cells. Okay, and there are different ways how to do the transcriptome analysis of memory epithelial cells, because that is the our in cell of interest. Okay. Okay, so once you know the memory parenchyma, and then I'm further going into the detail. I'm trying to explain how the cells are arranged in the cow as well as in the heifer, which is have not given any calf. So this is a typical cytostructure of lactating mammary gland, which is the single. This you can see. This is the alveolus, which secretes the milk, and this alveolus is lined with mammary epithelial cells, single layer of mammary epithelial cells. However, in heifer, there is no alveolus because it's not lactating, so they have called terminal ductal unit, TDU. So terminal ductal unit is composed of, so each here instead of 1, there are 3 and 4 different cell layers. Okay, so luminal layer, embedded layer, and basal layer, which is shown here. So these 3 layers is the basal layer, and then the cell, and then you have a luminal layer of epithelial cells because it's close to the lumen, and in between you also have a embedded layer of epithelium. So this is important to know because the stem cells, memory stem cells, present in these basal layer of epithelium. Okay, so once we know that, the localization of memory stem cells in the basal compartment. So please note that. Okay, so the question is, now you know where the stem cell, memory stem cell presence, and then the question comes, how do we know that, or what are the characteristics that people have been utilized to know the presence of stem cells within the gland. So morphological evidences, this early studies, 1997, 2002, and Bovine. So small light cells or large light cells, those have been described as the stem cells. So small light cells are described as the stem cells, and large light cells as a progenitor cells. Okay. Also, using flow cytometry, initial studies have shown the expression of site population fraction as a stem cell population, because those site population fraction was able to regenerate the gland when it was transplanted into the mice that was devoid of epithelium. So memory tissue of the mice, removing all the epithelial cells, keeping only the fat, and taking those site populations, putting inside the fat pad of the mice, was able to regenerate the whole mammary gland. So indicating that these site populations, that is coming because of presence of BCRP protein on the cell surface. So those are having these stem cell properties. The percentage of site population is 0.1 to 2% in human, 0.5 to 3% in mice, and 0.5% in bovine. So why I'm telling about this to you is, so that means out of 1,000 cells, the 2 cells will be the stem cells. So very, very rare population. So now you think about, if we want to utilize this for therapeutic applications, then this number is not sufficient. So somewhere, somehow, we need to expand. Okay, so that we'll talk about a little bit because And that is a complete different topic, how to expand those stem cells population. So third approach is the multi-parameter cell sorting, where use combination of different cell surface markers, either CD29 positive or CD29 positive high expressing cells. And this was the landmark paper that came in 2006 in the Nature, and then other group in human, they have identified these three markers as a marker of memory stem cells in mice as well as in human. And similarly, into the 2020 bovine in bovine mammary gland, they found the CD49F high positive cell not expressing the p cadherin, okay, marker. So that was characterized as the marker of bovine mammary stem cells. If you talk about what is CD24, it's a heat stable antigen. CD29, it's a beta-1 integrin. CD49F, it's alpha-6 integrin. Okay, so that means it's not uniquely present in only stem cells. It may present in different cell types also, but selecting one marker, two marker, or three marker would be better than selecting two marker or one marker. So more the marker you take, basically you're enriching the stem cell population. So simply taking one or two marker may not give the right amount of, you can say, enriched proportions of stem cells. And this is also evidence from the other studies, especially in mouse, because mouse is the most studied animal model in terms of the mammary stem cells. So these in this memory colony forming unit or forming colony, they forms the colony, and then you have a MRU, memory repopulating unit. And these two had the stem cell properties in mice. Using in bovine, this is the approach using flow cytometry. First, getting those live cells and taking those live cells, and out of these live cells, you take the CD45 negative and CD45 positive. CD45 is a marker of lymphocytes. So taking those CD45 negative population, you need CD45 negative population, you need this population, not CD45 positive population. So, and then taking this CD45 negative population, you go further, and then again apply those channels of different markers and identify different cell types. Okay, so in this lineage negative, which is CD45 negative, CD49F high positive because you have high and low, and then P negative, P cadherin negative population, and this is the true stem cell population. In bovine, that may not be applicable to other species. Please remember that. So, so the stem cells, it can, give rise to common progenitor cells, and then it gives rise to estrogen receptor positive and negative, again goes to the ductal cell and then alveolar cell, and then alveolar makes the secretory cells, and these common progenitor cells give rise also to myoepithelial cells finally. So that means, one cell give rise to 2, 3, and 4 different cell types, and also in the ductal you have positive cells, you have negative cells. So there are array of different cell types, though they look like all ductal cells, but phenotypically, immunophenotypically, they may not be the same. So that was the study using multi-parameter cell sorting to show the evidence of mammary stem cells. Also, we have a mammosphere where if you grow those mammary stem cells in culture, they form a ball-like structure. They're called mammosphere. and that dictates the self-renewable property, because if you chop that mammosphere and again reseed into the culture, again they form the second generation mammosphere and so on. And if you permit them to differentiate using the differentiation media, and those cells will differentiate into epithelial cells and myoepithelial cells, indicating that the stem cell has a differentiation properties. And this is a typical example of mammosphere, how it looks like. Okay, and this is things explaining, if you take the mammosphere formation, if you again chop them, and again it will form a new or second generation mammosphere. Okay, and this is showing you in all the species: horse, pig, cow, deer, dog, rhesus monkey, hamster, mouse, rat, human. They all mammary stem cells form mammosphere-like structure. Okay. One important thing here to show you that the shape, or you can say the morphology of the stem cells, may not be big in size, round in shape, large cytoplasm, large nucleus to cytoplasmic ratio. Sometimes even fibroblast-like structure can be seen. Sometimes we get puzzled how to, to say that this is stem cells. But believe me, if you grow them in culture, cells can take a different cell types, and that might be confusing. And then there is another study called label retention study. Due to asymmetric segregation of DNA, label is retained by the stem cells, and that give rise to label retaining epithelial cells. And that was my PhD study, where we identified identification of label retaining epithelial cells in bovine mammary gland. And likewise, it's not only restricted to epithelial tissue or mammary tissue, but also in the intestinal, muscle, and other species, mammary gland of other species. So if you want to understand how this label retention works, then you might have known this bromodeoxyuridine is a label, which is the thymidine analog. If you put that BRDU in the medium, then the cell, when it's replicated during the S phase of synthesis, it incorporates the BRDU. So that's how, so during the replication, BRDU goes into the nucleus or during the DNA replication, and followed by cytokinesis. So, the two daughter cells contain the BRDU, and those BRDU can be detected using anti-BRDU antibody using the technique of immunohistochemistry. And after that, then you don't provide any BRDU and allow them to divide without the BRDU. So what will happen after certain division? Those BRDU will get adjusted into the progenies. Okay. So if there is a random segregation of the DNA, then the BRD will get distributed into two daughter cells, and getting lighter and lighter, because in a subsequent division there's no BRDU now, so it will be half and half will be distributed to the progenitors, to the daughter cells, and finally it get exhausted. If that is a random segregation, but if that is a non-random segregation, which is a property of stem cells. So if the stem cell is getting BRDU, they do not segregate randomly. Okay, they go for non-random segregation. So that means the older strain that is having the BRDU retained with the stem cells, and then the other daughter cell, which is less, you can say, or which are differentiated in nature, the newly synthesized DNA is passed on to the progeny, and therefore those cells will not have the BRDU label. So therefore, the identification of BRDU label retaining cells is one of the method of identifying the stem cell population. And here is the study that I conducted. This is the typical structure of prepubertal terminal ductal unit. You can see the basal layers are blue in nature, and then the embedded layer and luminal layer. And in that, I'm showing you presence of two green color, and they are the representative of VRDU level retaining epithelial cells, which is shown on the right panel here. You can see those are the VRDU level retaining cells. Using the laser capture microdissection microscope, it's possible to dissect those level retaining cells along with the adjacent cells that are not retained any label. And likewise, we harvested four different populations of cell from 5 animals, and you do the macro analysis. And this is the differential expression of more than almost 600 genes within 2 cells. Imagine that. So within 2 cells, the difference between the label retaining epithelial cells and non-label retaining epithelial cells is almost 600 genes that were differentially expressed. Okay, and those genes were involved in the growth, proliferation, cell cycle, and all those. And with this, we have found HNF4A, which is stem cell marker, which is at the center of this network. And those genes which are red in color is upregulated genes, indicating that the stem cell network is operating in those level retaining cells. Okay, and this is the comparison. And finally found that the HNF4A network is operating in those level retaining epithelial cells, and which are present in the basal location, they are stem cells, and those which are present more apical location, and they are progenitor cells. Okay, and this is the typical representation of different novel markers of mammary stem cells found from the bovine mammary gland. And they are NR5A2, NUP153, FNDC3B, and HNF4A. So these 4 novel markers of bovine mammary stem cells have been identified. And here I'm showing you it's a role of these markers from the literature. So when we explore how these markers at expressing higher in those level retaining cells are really expressing in these cells, in the tissues, so that we can say them, okay. So you can see here that in heifer, the location of NR5A2 is basal, and also only few cells are expressing NR5A2. Similarly, the NUP153, again, the location is basal. FNDC, again, there's a basal location, and also the HNF4A, which is expressing at by few epithelial cells, and they are basal in nature. Then we also look whether that's the pattern in the cow or lactating mammary gland. Again, finding very few cells that localizing NR5A2, kind of 1 or 2 cells per alveolus, which makes sense. So that means these mammary stem cells or progenitor cells is able to replenish or to cell in these alveolus. So every alveoli has a 1 stem cell population, and that's responsible for tissue turnover at the time of milking. Okay, and this is the quantification, calf versus cow at peak lactation. Of course, at peak lactation, the expression of those cells were high, and one of the reason could be they are not only the stem cells but also the progenitor cells marker. So at the peak lactation, you all know the cell proliferation is very, very high. So that could be the reason that there are trans-amplifying cells are present, and so rapid proliferation is going on, and rapid cell turnover is going on at the time of peak lactation. And then slowly, if you go down to later lactation, say 120 days, 240 days, and then you can see the number goes down. And finally, at 300 days, you see that the percentage of those stem cells populations are very, very low. So this data is clearly indicating that the number of stem cell population at the late lactation is very, very low, and that could be, manipulated if you want to manipulate the lactation length of a cow. Okay, and this is the localization of 2 different markers. These are the localization of classical markers, which is people have done it. So I'm showing you just that. And then the new approaches to identify memory stem cells are the linear stressing experiment, where they tag those cells in the embryonic state and then they follow where they end up to. And those cells can be labeled in different colors using different tags, and that can be identified in later stage of the life. Okay, and so with that, one important discovery was they found that some of this memory stem cells are cyclic in nature, and some are not at all cyclic in nature. So that's the another puzzle came in 2017 and 18, and then later they give rise to a model. So initial I provided you the simple model. Now after this discovery, then found a new model. So there's two model. Model A say that during the embryonic stage, these activated memory stem cells are there, and its fate is getting decided before birth. So luminal progenitor and basal progenitor, and then when the animal is born or mouth pup is born, and then at the time the stem cells are quiescent in nature, and it's not going to do anything unless the hormonal signals or pre-puberty kind of thing come to into the force. Then those quiescent stem cell will come into action. Okay, so that's model A, and model B says no, there is no quiescent stem cells. It straight away goes to the luminal progenitor and then basal progenitor, and then it follow the differentiation pattern. So the later it was realized that there is a transition phase between the quiescent stem cells and proliferating memory stem cells. Okay, and these are the various markers to identify those quiescent stem cells and proliferating memory stem cells. Okay, so the division of stem cells, whether it is cells are dividing parallel to the basement membrane or perpendicular to basement membrane, dictate whether the epithelial cells or whether the ductal elongation will happen or cell will go into the for the milk production. So imagine that this is the basement membrane and this is the memory epithelial cell is dividing. So if the plane of division is a perpendicular to the basement membrane, then you see that there is a ductal growth. But if the plane of division is parallel to the basement membrane, then you will have a epithelial development. Okay? So kind of telling that the orientation of division of those stem cells, either in symmetric fashion or asymmetric fashion, is depend on the orientation of the plane of division. So see this in this example, in this type of where it's a perpendicular, you see that the stem cells which is dividing, it remain in the basement membrane. So these two will remain in the so the daughter cell will remain stem cells. But in the latter case, where the plane of division is perpendicular, then the cell is pushing to the more apical side, and the microenvironment of the basal location is no longer adjusting. So therefore, the stem cell will differentiate and becomes the progenitor cell. And again, if this progenitor cell divide in the similar fashion, then you will have a terminal differentiated cell. Okay, and these are the cells which are responsible for milk secretion. So the division of cells, symmetric, asymmetric, depends on the orientation of the division, and that decides whether a stem cell stays in the microenvironment of the basal layer or it goes away from the basement membrane. And that's how the fate is decided by the plane of And it was found that reprogramming of memory stem cell from basal to luminal and luminal to basal was possible using some transcription factor. So that means really that is happening. And this is the last one approach, which is the most recent one, is the single cell RNA sequencing, showing you at the, so this blue line is the time of birth. So you can see that this is the fetal memory stem cells. So whether it's a luminal in nature or basal in nature, the fate is decided before birth. And after birth is only the lineage commitment is done before birth, and after birth is just an expansion of the cell. Okay. Okay. So this is the last model where saying fetal stem cells is come into the transition stage after birth, where the alveolar to intermediate, to ductal to alveolar to lineage primed to induced memory stem cells can be interchanging. So stem cells can be induced to induced memory stem cells and give rise to different cell types, and this is the transition stage, and finally it gives rise to ductal or myoepithelial in nature. So at the time of need, like wounds, mastitis, tumorosis, okay, these iPSCs get activated. So initially they may be cushioned because it's not required by the system. So once it's need, they get induced. Okay, even the parity induces stem cell population. So parity induced memory stem cells, they have been reported in bovine, and that parity induced memory stem cells may be different from the heifer stem cells. Okay, so this is the latest model so far. That the fetal stem cells, and then after birth, the progenitor strips come where there is induced memory stem cells and a whole lot of intermediate cell types going on, and finally, at the time of pregnancy sets in and lactation starts, then they functionally differentiate into ductal or myoepithelial. Even in ductal, you have a luminal and basal. Okay. So this is all about the memory stem cells. So far, I think I have covered enough to introduce you what is the stem cell, how it looks like, how it characterizes, what is memory stem cells, how memory stem cells been identified using different approaches. Okay. Now coming to its application, which is most important. What is the application of stem cells? Let us talk about the productivity, because in bovine, that is the most important trait that we all consider. So stem cells can be utilized for chronic mastitis, treating acute mastitis, increasing milk production by enhancing tissue turnover. It can be used for dry core therapy, wound healing, and other inflammatory conditions. Those who want to read more, go through these papers: bovine memory stem cells, cell biology, meat production, agriculture, and again another paper, memory stem cells in memory health and milk production, and also memory stem cell expansion, memory stem cells expansion and animal productivity. So those articles will give you an idea how these memory stem cells can be utilized for enhancing production potential in bovine. So before I bring your kind attention to the lactation cycle, in order to understand how the manipulation of stem cells can be useful or can be possible to enhance the production. So I think it's logical to believe, and some of the data at the end I will try to show you that the manipulating those stem cell population can be useful in increasing milk production. So this there are three line graphs. So let us focus on this, which is in solid line. This is a typical lactation cycle in cow or buffalo. So at the peak lactation, which is somewhere here, so up to peak lactation, cell number is increasing. So during early lactation, cell number goes on, increasing and increasing, and you get more and more milk until you reaches the peak lactation, which is 90 days or 100 days in cow. So after 90 days, the production going down because of cell death. So here, cell number increase is less and apoptosis is more. Initially, apoptosis cell number in proliferation was more, apoptosis was less, and then after peak lactation, proliferation is less and apoptosis is more. So more cells are going to die. Okay, so that means when you come to the late lactation, the number of epithelial cells is almost half of the number that was present during the early lactation. Is unbelievable. So 50% of the mammary epithelial cells are dead at the time of late lactation in comparison to the cow during early lactation. So this is there in the literature. You can go to it. So my idea is, if we increase the proliferation during the late lactation, where the cell number, epithelial cells are low or down, apoptosis is more, maybe we can increase the cell number of epithelial cell number so that we have more milk, not in the same lactation, but of course in the next lactation. So therefore, dry cow therapy is very, very important. So dry cow 40 to 60 days are given. 60 days are too much. That's not necessary. As per the literature, 30 to 40 days is sufficient, provided that all those dry cow therapy have been followed. Okay, so if you don't provide the dry period, definitely the next lactation yield is going to be low, almost 20 to 40% low milk production. Okay, so during the dry period, the number of epithelial cells is approximately 50%. So if we increase the stem cell population during the dry period, then we are increasing the progenitor cell population. Okay, and those progenitor cell population, which is a trans-amplifying cells, will give rise to more number of mammary epithelial cells, and more number of epithelial cells, you all know, more amount of milk. So that means increasing the stem cell population is giving more milk because of more number of epithelial cells or more number of differentiated cell that came because of the cascades of proliferation from stem cell to progenitor cell and from progenitor cell to differentiated cells. Okay, so this has been well explained in this paper. So we performed an experiment in goat. So our idea was, if you infuse a compound called xanthocin into goat mammary gland, can we increase the stem cell population? And if we increase the population, can we increase the milk production? That is more important. Okay, so we conduct this experiment and we injected xanthocin into the mammary gland of a goat, one of the gland of the goat, and that was during the early lactation, from day 5 to day 7, and then milk recording was done, and that was a gland wise. So one goat was the experimental xanthocin treated gland, and other gland of the same goat was the control gland. Okay, and then we did the biopsy to know the stem cell number, transcripts, and also we recorded the milk. Okay, you can see that those are the important markers, like beta casein, estrogen receptor. This is differentiation marker MUC1, LALBA, and this is a stem cell marker was higher in xanthocin treated gland, and these are the markers we evaluate in both the glands. Okay, and we found that especially the star mark, which are highly significant in those genthicin-treated gland, and they are the milk fat synthesis gene, a milk gene, and also the stem cell marker like ALDH1, NRF-A2. Okay, so these were really increased in the genthicin-treated gland, indicating that genthicin was able to increase the stem cell number. And that was we examined how the genthicin is impacting those memory epithelial cells using RNA-seq, different pathways, and all those, which is discussed in these papers. Okay, so to summary, genthicin was able to increase the stem cell population when we measure the milk fraction. So it was significant different, but not very prominent. Almost it was 2% per gland or 4% per animal, which may not be very significant. And one of the reason is could be, we injected genthicin during the early lactation. However, if we injected during the late lactation, which we understand now, that would be beneficial rather than injecting during the early lactation. So maybe we like to repeat those experiments. Okay, and then that was the application of stem cell in increasing milk production. Now come to see the application of stem cell in mastitis, especially in chronic mastitis and acute mastitis. Okay, so these data I have taken from the literature. So in clinical mastitis and subclinical mastitis, clinical mastitis is only 5 to 10%, and majority of them are subclinical, and those are manifested by increase in milk somatic cell or decrease in milk production. Okay, and what happens during those mastitis? There is a damage of memory epithelial cell. Okay, it's a disruption, atrophy of alveolus, some because of lack of damage, and then there is a damage of blood vessels, no blood supply. So instead of say 5, now only 4 alveolus is secreting milk, and later only 3, only 2, and finally the gland is getting dry off. Seepage of blood into the milk, so you get rose milk, increased milk somatic cell count, which reduces the loss of milk quality, poor milk quality, decreased shelf life, and then the treatment of the mastitis is the antibiotic therapy. So the associated veterinary cost is there. So this antibiotic therapy has been practiced since 1940s. One advantage is that if you treat the targeted organism, then it will work. But if the antibiotic is not susceptible to those bacteria, then it's not going to work. So it's go waste. So the more disadvantage is that the presence of antibiotic residues in the milk, issues of AMR, antimicrobial resistance, and then those antibiotics, when we consume it, then disruption of gut microflora to the consumer, and also to the animal if the antibiotics is administered intravenously. So what the alternative therapies are? So alternative therapies are frequent stripping, and especially with oxytocin. If you give the oxytocin and do the stripping, remove the milk, then that's better. Herbal preparation that we all know, homeopathy that we all know, and then stem cell and its derivatives. So the question is whether the stem cell really works. Okay, we don't have much data. One data that I could found from the Peralta, which is reported in scientific reports in 2020. So this was the induced mastitis, injecting the streptococcus into the mammary gland, and then again injecting the stem cells at 6, 7, 8, 9, and 10 days. And then you see the data, and this is the colony formation unit on the y-axis in the bacteria, and on the x-axis you have number of days after treatment. So you can see that on 7th and 8th day, the number of bacteria was down. So the blue bar is the negative one, which was not treated with antibiotic, and then orange bar is the treated one, antibiotic treated one, and then the stem cell injection you can see is in between the positive and negative, indicating that the injection of these cells, I don't remember exactly, maybe 10 million cells per quarter, was able to reduce the number of bacteria, indicating antibacterial effect of stem cells. However, here it's 2.5 into 10 to the power 7 cells. So, however, the severity of the case, the number of cells injected, and the dose frequency of injection also matters to have a therapeutic effects. Okay. Likewise, in goat, a chronic mastitis was treated with adipose tissue-derived stem cells, and that restored milk production. So that means the stem cell was able to restore the functional restoration of the gland, and maybe fibrinolysis of those fibrous tissues, and helping the gland to restore into the milk secreting gland. So that was something encouraging to see. Other possible applications of stem cells, tissue repair, reducing inflammation, because the stem cells not only it has immunomodulator properties, so that means it reduces the inflammation, it repairs the tissue, regenerate the tissue, enhance the immune response, prevent the reoccurrence of the disease. Also, the growth factors and peptides has antibacterial effect, antiviral effect, and also has been shown to have a synergistic effect with the antibiotics. So that means, lesser use of antibiotics if you use along with the stem cells. Okay. However, there are certain challenges. First is the isolation and cultivation of stem cells, because they are very less in number. So you need to expand them in the culture without differentiation. So make sure that, the stem cell property remains there. Safety and efficacy, it should not be contaminated with any bacteria or virus. The safety is prime importance. Then regulatory approvals, I know, but pretty sure the government of India has no guidelines so far whether this has been done in the clinics. However, at research level, with the consent of the IAEC and CPC SEA, along with the owner's consent, at experimental level, this can be done. And then the cost and accessibility. So, the harvesting cells and culturing them, and then, maintaining them involves a significant amount of costs, and whether those can be reached to farmers' doorstep is yet to be yet to come, I guess. Okay, so I think thank you very much. So I encourage you all to download this app to know more about the stem cell in application in veterinary science. And if you have any questions, I'm happy to answer your questions. Thank you.
Thank you, Dr. Sundari, for very, very elaborative and very, very informative talk on stem cells and their use in mastitis and increasing productivity of livestock, so particularly in terms of meat production. So I think it has thrown adequate light on the prospect of using stem cells in future in veterinary therapeutics. I think it has benefited a lot to all of us. So I have a couple of queries, actually.
Yes.
Only out of your deliberations that I could understand that the destiny of the mammary stem cells, whether they become luminal stem cells or basal stem cells, is predetermined. And I think there is a possibility of inducting or inducing the process also, because the proportion of luminal stem cells prevalence in the memory tissues, as you have already rightly pointed out, that may be very more important for generation of user memory epithelial cells. So can you throw some light on the prospect of such kind of studies?
That's a good observation, sir. So far, we know increasing stem cell population can be helpful. And one good thing about those tissue-specific stem cells, which is a memory stem cells, is they divide by default, but they divide in asymmetrical fashion. So that means when there is a need, one stem cell will give rise to two stem cells. And if there is no need, then the stem cell will divide into one daughter stem cells and one differentiated cells. And then the fate of those differentiated cells, whether it will be luminal or ductal, will depends upon the need. So that understanding, I don't think any of us we have, but this is still in experimental stage. The study we conducted on goat and in USDA on bovine, that was increasing the stem cell population in vivo, that's within the animal, and then results was very, very helpful. But exactly to be pinpointing how to differentiate them into luminal or ductal, that is, I don't have that answer.
Okay. So another aspect that came to my mind regarding prospect of using stem cell therapy in chronic mastitis. I think you have already showcased some of the reports already in goat that
Yes, sir.
it has been tried. I think this is a big issue because fibrosis takes place to great extent in chronic mastitis, and
Right.
high-yielding animals, particularly in buffalo, as well as in high-yielding cows in Jersey or Holstein Friesian, usually become non-productive because of partial or complete fibrosis because of chronic mastitis. So I think there is a prospect of working more on this line so that we can replace the unproductive fibrous tissue by incorporating or inducing some productive, so to say, memory stem cells, so that we can regenerate the memory epithelial cells. What is your opinion on that?
Exactly, sir. That's a wonderful approach. I think we have the concept since last 6 years. Again, I'm taking that, reaching to the funding agency. So our idea was to first thing is how to get the stem cells from the cow.
Yeah.
So biopsy is the only solution we have, because slaughter is banned here, and alternatively, our idea was to get those stem cells from the milk. So few studies was there, that story I haven't touched here. So our idea was trying to get those stem cells, bovine stem cells from the bovine milk, and grow them and keep them in an undifferentiated state, and then utilize them for mastitis or chronic mastitis and see how that goes. So that's a very untouched field, to be frank.
Yeah, as you have rightly pointed out, that the major challenge is in isolation, identification, and cultivation of the memory stem cells. So, do you have any idea about any kind of work being carried out somewhere regarding induced memory stem cells? Whether we can convert any type of other stem cells to memory stem cells in culture? Is there any work going on in that line?
Yeah, you put me in tight corner, sir. I don't remember so far, because in bovine nobody is working. Very few lab in the world. You can all they are working in mice or human. In bovine no one is working. Our lab is working. When I was at USDA, So my advisor lab was working. So, and then in Sweden, there is one group, and then one more group, one DVAL in US. So they are working, but not in this aspect. So now they are working instead of inducing mammary stem cells, they're working on the growth factors and secretory factors secreted by the mesenchymal stem cells, and that to be used as a drug. Okay, we are also working towards that line. So that means, injecting a stem cell might be, seems like kind of unethical, but if you inject some kind of, proteins or peptides that are secreted from the stem cells, and those are natural and acellular product, a sterile product, that will be more application rather than injecting a stem cell itself. So we are working on that, sir.
Thank you. Thank you very much. My last question will be, you said about the ability of xanthocin to induce the proliferation in memory stem cells. So what may be the possible mechanism of action, how it can induce the proliferation of the stem cells? Any literature that you have?
Yes. So that the concept, sir, came in 1997. At the time, my collaborator, Dr. James Shirley, he was working at NIH. So he introduced that concept in human. And then when I was doing PhD, I took that and tried to extrapolate those using bovine mammary stem cells, and we expanded that in vitro first, and then in vivo within the heifer. So the possible mechanism so far we know is that, so the xanthocin, when it goes into the system, it forms the xanthocin monophosphate, okay? And so the xanthocin to xanthocin monophosphate, and then you have the xanthocin monophosphate is, if the concentration of XMP is more, then cell divides symmetrically. And if the concentration of these ribonucleotides are less, then cell divides asymmetrically. So the xanthocin straightaway goes into the nucleotide pathway and forms the xanthocin monophosphate. In nature, this xanthocin to xanthocin monophosphate is a rate enzymatic step. It's called xanthocin monophosphate dehydrogenase. Okay, so when you inject the xanthocin, it circumvents that pathway and straightaway increasing the xanthocin monophosphate concentration within the cell. So once the xanthocin XMP concentration is more inside the cell tends to differentiate, tends to divide symmetrically. That's why. So, and there are a couple of genes that are responsible that goes up and down. Yeah.
Thank you. Thank you very much for the nice explanation.
The session is open for discussion. If the students or participants have any query, so you can ask it.
Anyone from the audience having any query?
ये स्टूडेंट पीएचडी के हैं, मास्टर्स के हैं, कौन है सर?
Yes, PhD and master's students are faculty.
Sir, it is really very informative lecture. Thank you very much. And I have two queries. First, you mentioned that 3 marker is better instead of 2 marker to identify the stem cell. So what may be the reason behind this?
Okay. Okay, so so one marker, that genes are polygenic in nature. One gene controls many traits, right? So one marker, not one, those any of the marker, stem cell marker, not exclusively expressing on stem cells. It expresses on many different cells. Okay, so say selecting CD45. Okay, CD45 negative or CD45 positive. So basically, you are selecting only lineage, those that are hematopoietic in nature and those that are not hematopoietic in nature. If you include one marker, say. Now you selected say I don't want hematopoietic in nature because it's a mammary stem cells. It doesn't come from the blood. It should come from the mammary tissue. So I will take CD45 negative, right? Then you select another marker, say CD44. So CD24. So CD24 again is a heat stable antigen. It may express on many cell types, okay? And then CD44 high or low. Okay, I want CD44 high. So take CD44 high. Okay, so it transforms 1 lakh cell, then you have a 10,000 cell CD45, say negative, and out of those 10,000, 5,000 was CD44 positive, and then you include one more marker. Okay, and then from 5,000 it comes to 1,000. So likewise, you increase more and more number of cells, a marker, you are basically enriching the true stem cell population. Remember that the stem cell population is only 0.52, 1% or 2%. So that means only one stem cell is there out of 1,000. So in 1 billion, you will have only 100 or 1,000 cells. So increasing more marker is better. But now the technology has come, it's called kinetic KCS, kinetic stem cell counting. So where it doesn't require any marker. Okay. So these marker is sometimes on and off depending upon the physiology of the animal, age of the animal, okay, species of the animal. So that may go on and off. So mouse memory stem cell marker may not work in bovine. Bovine stem cell marker may not work in buffaloes. Okay, but the kinetic cell-based counting, which is a level-free marker, markerless selection, it based on how the stem cell divide, how much is population doubling time, and all those. And there is an algorithm. You can go to asymmetrics.com if you want to learn more. And that's there in my app. So you learn that, then you will understand. Okay. So many more things are there. So this is only a glimpse of story.
Okay, thank you very much, sir. And one more query is there. So you talk about that inducing some molecule to increase the population of stem cell.
Yeah,
And
gentle.
so second approach to injecting the stem cell SS. So which means procedure may be the better for means in vivo or in vitro experimentation as per your suggestion?
You said one compound, I said genthicin, that increases stem cell population. And what was the other question you said? Other compound?
Other compound I'm not talking about. So injecting some molecule, if we could able to increase the stem cell population, and same thing if you put the cells as such the stem cell. So which approach may be the better as per your suggestion?
You mean to say in vitro and in vivo?
In vivo and in vitro.
Place of genthicin in vivo and in vitro. I think he is asking that question.
No, he is telling replacing the cells by directly
Directly.
making a replacement of cells, intact cells. One approach is injection of certain molecules so as to increase the proliferation rate of the stem cells. Another approach he is telling that is just replacing or implantation of cells, stem cells.
Oh. Yeah, so injecting stem cells will not replace the stem cells.
Yeah.
Because say the classical experiment have done, so the exogenously administered cells were labeled with some marker just to trace where they are, how they are lodging within the tissue, where they are going, and they couldn't detect those labeled cells within the tissue, so indicating that cells are not lodging or residing. Any foreign cells will not go and lodge within the tissue. हम ऐसे बर्दाश्त नहीं करते South Indian वाले North India में रहे, North Indian, some kind of things are going on. Just an analogy I'm saying. So no, it's a very less chance that cell will survive and replace or start repopulating. Yes. So one of the mechanism of action is, which is my second proposal of DST, State University Research Excellence Grant. This stem cell will die and becomes an apoptotic bodies. And those apoptotic bodies, once you inject the stem cells, they're going to die. and those some may reside and die later, but many of them are die, and then they become apoptose. And those apoptotic bodies are engulfed by the macrophage and starts the cascades of cytokine events. And those cytokine events is giving the paracrine effects, and those effects draw the expansion, or you can say the therapeutic response given by the stem cells. Make sense?
Yes, sir.
Yeah,
Yeah,
thank you.
thank you.
So any other questions from the students?
Good questions. Wow.
Dr. Chaudhary,
Sir,
so what is the chances of establishing stem cell culture from biopsy material?
from which material, sir?
Biopsy material from the order.
Yeah, possible, sir, possible.
And whether IAC gives permission for multiple sample collection?
Permission is required, sir. That's why I've said, we are thinking to get it from the milk. But so far, I'm getting contaminations the last 4 months. We have some data, but not something that I can show you here. Biopsy is permissible, but the moment you approach to any of the farm, they say, these are the elite animal, biopsy is not possible. Straightforward, right? And the more other issues are, the once it is, they do the biopsy, they're going to be mastitis because, the sanitary conditions of the farms, and then our management is not up to the mark. Irrespective, in the US, remember one experiment we did, in one cow, we performed 6 biopsies. 1 quarter, 3 biopsies, 3 hour, 6 hour, 12 hour. And on another quarter of the same cow, another 3 biopsy, and without antibiotic, no mastitis.
This is the level of yeah.
Yes. And that was a drill biopsy, not means, taking the whole drill and taking the 2 or 3 grams of tissues each time. Amazing.
So I think the population of stem cells goes down drastically towards the, you can say, completion of 4-5 lactation. Is it like that?
Yes.
Because we tried to establish some culture from the mammary gland here, because in the Ceylon Meghalaya, there are a lot of animals.
Mm-hmm.
I could successfully establish culture in your project when you were outside US, one of your students. So only once I could successfully establish a culture from mammary gland. But here we tried from around 4 to 5 times from the slaughterhouse animal. However, we could not establish any culture.
Yeah, so that could be the reason, sir. The number of cells are really low, especially when the animal is dry.
So it's really very tough work, but you people are trying to do some good work. It's really appreciable. Any questions from the others? Yes? Yes. So if there is no one, or you can mail also to Dr. R.K. Chaudhary. It is available in the.
Please download the app. You encourage, sir. They should download and read it. So good.
So he has developed app also, so you can get a lot of idea about the stem cell and the application through these apps. I'd like to request Dr. Louis Moni Porkalta to give a vote of thanks.
Thank you, sir. So on behalf of the organizers, we'd like to thank Dr. R.K. Chaudhary for sharing his immense knowledge in this session. So we are really thankful and also anticipate similar kind of association in days to come. So we also like to thank Director of Research and former head of the department, Dr. Prabodh Borasa, for being here in this session in spite of his really busy schedule. So I would like to thank all the participants who have joined from different parts of the country and to be a part of this session. My thanks also goes to all the members who have joined the session online sitting here. We will also like to thank the Government of India for generously funding different activities of the department, including this project under which this particular online session has been organized. So I would also like to thank the administration of Assam Agricultural University for giving permission to organize this particular session, and also making all the physical facilities available. Thank you so much once again. Thank you.
Thank you very much, Dr. Chaudhary.
Thank you, sir. Thank you, dear sir. Thank you.
Oscar,
Really nice to see
Namaskar.
you here.
see you sometime again.
ठीक है सर, मिलते हैं सर.
Okay, thank you very much.
बाय.